Repressing. So let's discuss a very important example here. We cannot discuss all 4075 or so um, biotechnology pharmaceutical drugs here, but we will discuss some key pharmaceuticals. First one is human interferons. Interferons, look at their name carefully. Interferon indicates interference. They interfere. They are the signaling proteins. They are basically glycoproteins in nature. Glycoproteins are adjunct proteins. They are combination of glyco and proteins. Glyco is the carbohydrate portion, and proteins, as we know, is an organic molecule. So when we combine carbohydrates and proteins together, they make an adjunct known as glycoprotein. You might also be familiar with another adjunct known as proteoglycan. Proteoglycan is also made up of proteins and carbohydrates. But although the nature of the two is same, like they are composed of carbohydrates and protein, whether it is glycoprotein or proteoglycans, but they are different. How they are different? They are different in the composition. For glycoproteins, the protein is in majority and glyco forms a smaller portion of it. Whereas for proteoglycan, it is the carbohydrate portion which is bigger in size, and the protein portion is smaller. So these glycoprotein interferons, they are the signaling proteins which are produced by the cell when it encounters viruses or when it becomes tuberous, means when it becomes cancerous, or because of other immuno challenges. So when this the cell which becomes infected by virus, then it will start giving the signal, and the signal is in the form of interferon. Interferon will modulate the immune system. It will help in activating the antiviral defense, and it will also regulate inflammation, which actually is a hallmark when some immunogenic challenges are being uh, put forward to any cell. So the interferons are of three types: type 1, type 2, and type 3. So type 1 include interferon alpha and beta, which are involved in the antiviral responses. Type 2, which is interferon gamma, it regulates the immune system activities. And type 3 is interferon lambda, which protects epithelial cells. And they are encoded by different genes. For example, if we look at interferon alpha, it is encoded by a family of 13 different. They are different, but at the same time, they share regions of similarity. When it comes to interferon beta, it is encoded by two genes, and interferon gamma is encoded by a match strategy. They engineered interferons with combined properties based on different members of the interferon alpha gene family, and because they have different extents and specificities in their antiviral activities. So I will show you with the help of this figure. They made interferon hybrids, and how they did it, you can see the two genes interferon alpha 2 and alpha 3 on top. Alpha 2 is in teal color, is shown in teal color, whereas alpha 3 in the peach color. So you can see the presence of restricted enzyme sites on both of them. So they are subject to restricted endonuclease digestion, but that digestion was kept partial. So with the help of partial restricted enzyme digestion, they generated overlapping fragments, and then those fragments were combined to make the hybrid genes. So you can see here alpha 2 portion is combined with the alpha 3, and the same is true for the rest of the hybrids as well. So then these hybrids were later checked for their potential therapeutic property, and the successful ones which showed promising results were then picked up back in 2003, and then they were subjected to clinical trials, and after the successful clinical trials they are approved for the use as human therapeutic agents. You can also see another figure here for the construction of hair hybrid interferon alpha genes, and here multiple genes are used. You can see four genes here because they are these are shown in different colors. First they were amplified with the help of PCR, and this amplification was done on the target cDNA rather than amplifying the gene from genomic DNA. They used the complementary DNA, which is developed from messenger RNA. What is the idea behind? The idea is to remove the intronic portion so as to make the gene size compact, and to avoid post-transcriptional and post-transcriptional modifications of the messenger RNA, which cannot be done within a prokaryotic host. So, after PCR amplifying the interferon cDNA, they were partially digested with DNAs, and the random cuts are generated, and then they are rearranged in a mix and match manner. And as a result of this hybrid formation, now we will call them as chimeras, because here we are not talking about two different sources, rather we are talking about multiple different sources which are combined. So chimeras formed, and those chimeras are then later used for expression purposes. Here you can see in the yellow box the resultant interferon alpha gene shuffled libraries were first constructed, which we have already discussed the libraries earlier in the course of this health biotechnology. So you are aware of what libraries are. So after the construction of libraries, those libraries were tested for the anti-proliferative and antiviral activities of the interferons, and those which gave higher values were then used for expression purposes, and those hybrid fusion interferons were then used for marketing and therapeutic purposes. So you can see here, and now there a concern which was there in the market, uh, and the concern was the short life of interferons. Interferons are used in hepatitis C therapy as well. We already are aware of hepatitis C virus infection, which is one of the most common cause of liver diseases, not only in the developing world but also in the developed world. But I must say that the number of patients affected with hepatitis C is much more higher in developing world or the poor world than the developed world, and the reason is the poor sanitary conditions in the developing and the underdeveloped world. So you can see that hepatitis C virus affects nearly 200 million people worldwide, and it may develop into liver cirrhosis or hepatocellular carcinoma. Almost a decade ago, this virus infection was considered as deadly or terminal disease, and there was not no complete cure was not available for it. But with the use of interferon for controlling the hepatitis C infection, Now hepatitis C can be managed. Alhamdulillah. So you can see therapeutic agents that maximize early antiviral response and also maintain the viral suppression is what is needed for uh, controlling infections like hepatitis C virus. So, the effective treatment of hepatitis C included the combined use of antiviral ribavirin with interferon alpha. But in order to make it a success, longer acting interferons were needed. Why? To minimize the side effects of the interferon treatment, to lower the required dose, and to decrease the required frequency of the treatment. Now I am concluding this talk here, and I will soon share you the rest of the talk.